This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1233, recorded on the 4th of July, 1776. No, not really. It's 2025, but that's why it's a holiday today in the U.S. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here, and it is a gorgeous 4th of July here in Western Mass, 81 Fahrenheit, 78 Fahrenheit. Sorry, it just updated 26C. And it's clear blue skies and light breezes and just absolutely lovely.
28C here.
Same there.
Sunny. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. I have a special weather report for you today. We are experiencing the remnants of the tropical storm that came up through the Gulf of Mexico and glanced off Mexico and landed in Texas. Okay? And that's both good news and bad news. The good news is that the temperature here is, I'll bet you it's close to a record low, 78 degrees. And it's been raining here in Austin for over 24 hours, which we desperately need, and soaking my garden, which I just love. The bad news is that about 100 miles west from here, and I think you will probably read about this, it's serious rain. They've gotten like 14 inches in 24 hours, and it affects the Guadalupe River, and in particular a town called Kerrville along the river. There's been significant flooding that affects both commercial and recreational properties. An RV park has been wiped out. A kid's summer camp has been wiped out. It's not been tallied yet, but there are going to be deaths involved in this and very, very significant damage. So my heart goes out to those folks. And here we are.
And if you're downstream of that, do not drive across any dry riverbeds.
Yeah, absolutely. Well, people in Texas pretty much
know not to do
here.
That. Yeah,
they know not to do that. Yeah.
Why it could fill up right away?
Yeah, you get these. We have a lot of dry riverbeds, arroyos, really. Okay, and a lot of them actually that typically flood will have flood gauges, depth gauges next to them, and gates that close when they're in danger of flooding. So, but it's not good in Kerrville.
And on back roads in Texas and other parts of the Southwest, sometimes there will be a road that does cross a dry riverbed that's usually dry, and you always have to be very careful when you do that.
If you see water
Look
there,
both ways before you cross the riverbed.
Wow. Well, if you like these programs, these science programs, we give you real science, no lies, no misinformation. We'd love your support. We don't do ads. We just depend on your donations for our costs. Go to microbe.tv/contribute. So today is, as you heard, it's July 4th. It's Independence Day in the U.S., and I call it No King's Day.
Yes.
Because originally we were celebrating the independence from the King of England, and today we would rather our president not become a king. No King's Day, although he's working very hard to do that. I have a quotation for this day, and it's attributed to William Pennepacker. Dear America, you are waking up as Germany once did to the awareness that 1/3 of your people would kill another 1/3 while 1/3 watches. And I don't do quotes regularly like like Daniel does, but that's a good one for today. No King's Day. We should update the name.
Yes.
To No King's Day. Little news about ASV coming up July 14th through the 17th. It'll be hosted by McGill University and held in Montreal at the Palais des Congrès de Montreal. ASV.org/ASV2025. Two reminders from Kathy Spindler. Remember that one of ASV's sustainability initiatives is to reuse the name badge holders. See if you can find yours and put it with your suitcase so you don't forget it. And number 2, email reminders have been going out with helpful things. If you have missed those, they're posted on the meeting homepage. They're labeled announcements. Standard registration is over June 30th, so now rates are higher. So hopefully you've all registered. Now,
is it still cheaper to register in advance than to do it on the spot?
I think so, yeah.
Okay.
Wait, is it cheaper to do it in advance? Yes.
Yeah, like to do it if even though you missed the low advance registration rate, is it cheaper to do it now than to just show up at the meeting and do it there?
I don't think there's any difference, no.
Okay.
We're going to have a TWiV meet and greet on Wednesday, the 16th of July, 9 to 10 p.m. in room 522A. This is during the social hour. I'll be there with Angela and Kathy.
Cool.
Wednesday, July 16th. All right, we have some news for you this week. One article in the New York Times, unfortunately, I'm sorry, paywalled. And it's paywalled for me, too. The top FDA official overrode scientists on COVID shots. So I will summarize it because I remember this article. So the head of CBER, Center for Biologics Evaluation and Research, this is the division of the FDA that deals with vaccines and biologicals, I believe, is headed by Vinay Prasad, who is a oncologic surgeon. Okay, and he was advised by people at FDA that we should continue the current recommendations for COVID shots, and he said, "Nah, I don't think that they cause myocarditis. I don't think pregnant women and children and people under 65 should get them." And they said, "But there's no more myocarditis, and in fact, the myocarditis is worse if you get infected. So if you want to prevent myocarditis, you should be vaccinated."
Yep.
He said, "Nope, I don't trust it." So now we are in an era where the experts don't prevail. He's not an expert on vaccines, right? He's an oncologist. And while he may understand science, he's got an agenda. So it's very disturbing. Get used to it, I guess.
Yeah.
He wrote, quote, "Even rare vaccination-related harms, both known and unknown, now have a higher chance of outweighing potential benefits in non-high risk populations." Okay, the experts disagree.
He just made that up.
There's no evidence for that. The evidence is to the contrary, that the risk-benefit analysis of the COVID shots favors their use.
And his argument is essentially there might be unknown risks we haven't discovered yet, which is preposterous in vaccines that have been used now in hundreds of millions of people, possibly billions worldwide.
Precisely. Okay. An article in Nature, I think this is open, by Mariana Lenharo. NIH-funded science must now be free to read instantly as of July 1st, which is a couple days ago.
Yep.
So if your research is funded by NIH and you publish a paper, you have to give the public access to the paper. So the journals are not going to change their paywalls, but you have to post this paper somewhere so that people can see it. And this was originally established under Biden, and it was supposed to happen in December of this year, and Trump has pushed it up to July 1st. Thoughts, Alan?
Well, this has been in the offing for a while. Everybody saw this coming. It's actually been a trend for years. There was a policy the NIH put in place a few years ago that NIH-funded research had to be made available for free 6 months after publication. And then, they've encouraged open access publication on and on. But it's been very clear that this is the trend, that this is where things were headed. Now, by by pushing the deadline for it back by almost 6 months, Trump has, of course, created chaos, which is his brand, because everybody was expecting this to go into place December 31st, the end of this year, and now all of a sudden, here it is. So now everybody has to figure out, everybody who still has an NIH grant that hasn't been pulled out from under them, has to figure out how they're going to publish their research and make it available publicly for free. The journals are basically hosed. I don't know how this is all going to play out.
So they say you can put your paper at PubMed, but the problem is the journal still has this 6 to 12 month paywall policy, and they don't jive, right?
Well, right, but if PubMed Central has the paper and people can access it there, really most people find papers on PubMed, right?
Yeah.
And so they're just going to click on the free access link and not go to the journal at all.
Right.
So for the folks out there who are not familiar with academic publishing, and Alan can fill in and correct me on this, it's not like your grandma's publishing that you're aware of, where the publishers make money by selling their product and even pay the people who write their material for them. In this particular case, the publishers can make money by selling subscriptions to their product, mostly to academic institutions and et cetera. Depending on the journal, there can be a fair amount of private customers. But the people who are publishing the material don't get paid. They actually frequently pay the publishers to publish their stuff. So in that case, the publishers can make it two ways. And that's kind of the model, is that the person who's going to publish somehow pays for publishing it, either in page charges or otherwise. And so now the question becomes, if I have to publish my article, make it available free of charge, who's going to pay the publisher? And how's that going to happen? Because, now they don't have control over it. And by the way, this all gets done through PubMed. Mechanistically, I don't think it's all that difficult. But the cash flow here is now all screwed up. All right? We're halfway there because we've been dealing with, as Alan said, for a while. But the ultimate solution, I don't know how that's going to work out. This article is good because it goes through all the possible twists and turns.
And really, a big part of this is that the way the scientific publishing industry evolved, certainly by 20 or 30 years ago, it had turned into kind of a scam, largely thanks to one publisher called Elsevier that maximized its value by jacking up its subscription rates to institutions, because they realized that research institutions have to buy these subscriptions. And so they negotiated deals with academic institutions for extremely high subscription rates for all these journals, made massive amounts of money off a product that is, as Rich explained, effectively being produced for them for free. The researchers do the research, they write the paper. The peer reviewers, who are unpaid, are other researchers who read and review the papers. The entire editorial board is unpaid. You have some small office staff that's responsible for triaging the papers and sending them out to reviewers. You have to pay them. But when the main content is being produced for free and you've got, just this little office staff, and then you're charging ruinously high subscription fees, you can make a heck of a lot of money that way. Problem is, eventually the people you're making the money from get a little sick of it, and that's where the open access revolution came from. And, here we are.
Scientists are such suckers. We do all this stuff for free. So, basically, I am fully in support of the notion that federally funded research ought to be open access. It makes perfect sense to me. But the publishers do need to make a little bit of money, okay? They at least need to cover their costs, okay? Maybe there ought to be, well, actually, hasn't Trump proposed essentially a federally funded, it would be nonprofit, publishing machine, right? You know?
Yeah.
I don't know. I haven't thought that one through.
Yeah, and the open access journals have made their money by page charges. As Rich explained, you pay to publish your stuff, and then everybody can access it for free, and that's paid for out of your grant money.
If you have a grant.
If you have a grant. So the open access model has actually been very successful, which is why all of us, I think, probably get massive amounts of spam from scammy open access journals. Submit your papers to us. But there are legitimate open access journals that, have made quite a lot of money, PLOS One being the big daddy. This is the system that has been built. There's also there's a problem that I've been railing about for years that nobody ever pays any attention to, as far as I know, which is archiving all this stuff. Because now that it's all gone digital, and that, by the way, is why we're able to have all these open access journals, you don't have printing charges, you don't have mailing charges, so the costs went way down. But essentially, all this stuff is being put online in whatever the format du jour is, and we're just assuming that it'll all still be there 20 years from now when somebody wants to look these papers up. And I don't know how many people out there have had this problem where you've got some old file on your computer and you can't open it anymore. Now magnify that by a few billion and you've got the scientific publishing enterprise, and this is what we're doing with the scientific heritage of mankind, but nobody seems to be concerned about that amidst all this profit stuff.
So a related story, which I just thought of and I put in, is a science article. Trump cuts subscriptions to Springer Nature journals. So the administration has ended some agencies' subscriptions to Springer Nature, which includes Nature, and 3,000 journal titles. And it's not clear if NIH is involved, but it is HHS, so it should be NIH too.
What's the rationale behind that one?
Because the journals are corrupt.
Oh, okay.
They published they published something he didn't, some fact that he disagreed with or whatever. And if I were going to pick on a publisher and I were president, this is not the publisher I'd pick on. But anyway.
Yeah. I mean, the three of us were raised in this publishing system. And while it's not perfect, it does work to disseminate science. And
full disclosure, throughout my career, I have made a significant amount of my money from writing news for these subscriber-based journals, including some of the nature journals. And they were paying me out of the subscription fees that they were collecting. And, we can argue about what the profit margin should be, but there is, I like to think, some actual value being added.
I think if you're a long-term TWiV listener, you'll recognize that we've been talking about this for 15 years.
Yes.
Okay, one other story, two stories from Reuters. GAVI, the Global Vaccine Foundation, Vaccine Alliance. Remember, last week we reported that RFK Jr. was withholding the U.S. donation of $1.3 billion. Well, they've made that up. The U.K. and Gates have made up that $1.3 billion. They're still a little short of their goal. They got in $9 billion. They wanted 10 or 11, I think. But at least the U.S. shortfall has been taken care of. I think that's good.
Thank you, Bill.
Yes.
Thank you.
Yes,
UK.
thank you, Bill.
Yeah. Okay, our last story is from CIDRAP. USAID defunding could lead to 14 million deaths worldwide from infectious diseases by 2030. This is based on a Lancet article where they model what effect the withdrawal of the U.S. from USAID has on infectious diseases caused by a number of agents, mostly in children, including HIV, tuberculosis, malaria, malnutrition, diarrheal diseases, and so forth. So they say If we don't restore the cuts, then we're going to have a staggering number of avoidable deaths, basically 5 years.
Yeah. Yeah, and it's a human tragedy, and it's a geopolitical disaster strategically for the U.S. I mean, even if you look at this in totally cynical terms, when nations have these horrible health outcomes that are eating away at their economies, wars come from stuff like this. And this becomes our problem, regardless of whether we want to think of it as our problem.
Exactly. As Daniel Griffin says, no one is safe until everyone is safe.
Yeah.
Just incredibly short-sighted.
And tens of millions of people. Just appalling.
Okay. On to the science. We have a snippet in a paper for you today. Our snippet is a paper in Cell Reports. The SARS-CoV-2 trigger highlights host interleukin-1 genetics in Epstein-Barr virus reactivation, mainly from a group of institutions in the Czech Republic.
Yes. Yeah, this is a well-checked paper. So yeah, it's first author is Petra Schneiderova, senior author is Eva Krigova. Group is from University Olomouc and University Hospital Olomouc, Czech Republic, VSB Technical University of Ostrava, Czech Republic, Institute of Molecular Biology in Yerevan, Armenia, and then Masaryk University and University Hospital in Brno, Czech Republic.
So Epstein-Barr virus is a herpes virus, which infects 90 to 95% of the adult population and establishes what we call latency. The genome is present, but no infectious virus is produced. And then periodically, the genome is reactivated. It switches from a latent to a lytic cycle. If you make virus, it can spread to other people, and it can also cause some diseases. And, there are many... We really don't understand all the implications of reactivation of EBV, and we don't know what causes reactivation, in contrast to herpes simplex virus, where we have a pretty good idea. Stress, UV light, sunburn, all that stuff cause reactivation. We don't know for Epstein-Barr virus.
And EBV reactivation can cause clinical symptoms, or, if I understand correctly, it might not, right?
That's right.
Yeah, that's correct, yes. Did we already say 95% of the population has this virus?
Yeah.
And did we mention that? Sorry. One of the most familiar diseases caused by EBV is infectious mononucleosis.
Right.
And there are several. It establishes latency. The primary site of latency is B cells. So that's a lymphoid target, part of the immune system. So no surprise that there are several diseases that involve lymphoid tissue. Another notable cancer that it's associated with is Burkitt's lymphoma. And there are a few others.
Yeah. And so people have been, one of the problems of studying reactivation of EBV is this combination of some cases will be symptomatic when it reactivates, some won't be. And also, we don't know when it was reactivated or how, because it happens in different people at different times. But what this paper is doing is taking advantage of the kind of unfortunate natural experiment of the COVID-19 pandemic, because SARS-CoV-2 infection can apparently sometimes reactivate EBV, right?
Yep. So
the other one I should mention briefly, people can go back to the episode from Croatia, the European Congress of Virology, and we've talked about this before several times, but the EBV is now strongly implicated in the etiology of multiple sclerosis.
Right.
So the idea in this paper is you take a large cohort of people, most of who have EBV, latent, right? And then you look at a trigger that is associated with EBV reactivation, which is infection, and in the case which Alan mentioned already, infection with SARS-CoV-2. And then they further look at long COVID symptoms to try and see if they're all related. So long COVID here is defined as a infection-associated chronic condition that's present at least 3 months as a continuous relapsing and remitting or progressive disease state that affects one or more organ systems. It happens in over 10% of people after infection with SARS-CoV-2, and you get a lot of symptoms, right? Shortness of breath, fatigue, chest pain, cognitive impairment, memory lapses, fever. Over 100 million people have been affected worldwide with long COVID. And so we want to figure out what causes it so we could treat it. There have been some mechanisms proposed to be responsible as triggers for long COVID, including Epstein-Barr virus reactivation, SARS-CoV-2, persistence of SARS-CoV-2, autoantibodies, type 2 diabetes. Those are main risk factors for long COVID. But they point out that in most studies, the patients self-report symptoms through questionnaires, and this is not as good as having a diagnostic test. And so we're going to try and do that in this paper, as you will see. So they have a cohort of adults that have had the same trigger, SARS-CoV-2, and they're going to look at EBV reactivation and post-acute outcomes. And they also look at the role of interleukin-1 and interleukin-10, two cytokines that are produced for which there is some other evidence that these might be involved.
And one of the advantages of using the SARS-CoV-2 trigger in this is these are people who have a laboratory-confirmed diagnosis of SARS-CoV-2 infection. You can pinpoint when that happened, sometimes to the day, but at least within a short time period. And they have samples, I guess blood samples from a control population before the pandemic.
Right.
So they can do controls who were never exposed to this virus at all, and then they can do their experimental group is these people who have a specific timed exposure and then long COVID.
So they have 687 individuals who basically went to pulmonary centers for long COVID symptoms that lasted at least 4 weeks to 4 months after SARS-CoV-2 infection. So at the time they were examined, 84% of the individuals had clear pulmonary pathology on X-rays or impaired pulmonary function tests like vital capacity, expiratory volume, and so forth.
So their lungs are messed up.
They have clear lung problems that you can document with tests, right?
And quantify.
It's not them saying, I can't breathe.
And an important part of being able to test it is you can quantify them. You could say this is more severe than that. Whereas the self-report, you got lung problems, but to what extent?
But they also have self-reported information from these patients. 70% of them had subjective symptoms on questionnaires like persistent cough, fatigue, etc. And so of these individuals with long COVID, 76% had a mild to moderate course of COVID previously. Men were more likely than women to present with a severe to critical course of COVID. And up to 60% of all these individuals in this study had at least one comorbidity. And the most common ones were arterial hypertension, hyperlipoproteinemia, and diabetes. Interesting. So they look first at seropositivity for EBV, and 98% of the individuals in this cohort were seropositive for EBV. That's not surprising.
That's the population. Yeah.
That's the population number, They also look at antibody. So they use antibody to EBV as a measure of reactivation because when you're in the latent phase, your anti-EBV antibodies are low. When you reactivate, they go up, right? So that's their measure of reactivation. So they look, of course, at IgM, IgA, IgG in these patients. They look in the COVID naive group. They have a control group, as Alan said, people with long COVID and people hospitalized in the ICU in the acute phase of COVID-19. So 3 different study groups. They're looking at antibodies, okay? So when they look at COVID naive controls, the reactivation is similar to that in patients with long COVID. But there was a trend to higher EBV reactivation in the patients in the ICU.
This is what I had problems with. Okay, so let's just emphasize this and keep it in mind. The SARS-CoV-2 naive and long COVID people have a similar profile relative to EBV.
Right.
That doesn't necessarily indicate active reactivation, but people who are in the hospital dealing with COVID, they score positive for EBV reactivation. Got that solid? Is that right?
Yes.
Okay.
Yes.
Well, they do have reactivation in the naive COVID controls and people with long COVID. It's 27%.
But they're the same.
They're the same.
And it's 27%, right? But it's 37% in the ICU patients.
It's higher in the ICU
fine.
patients.
It's higher in the ICU patients.
And we don't know, I mean, in the ICU patients, it might be all the other stuff that's happening to their bodies in the ICU and not SARS-CoV-2.
So as you implied, if not stated early on, Vincent, one of the difficulties in this study is the background is so high for all this stuff.
Yeah.
Because we're crawling with EBV and probably reactivating all the time.
Yeah.
I mean, it remains, even after the study's over, it remains an issue, right? You cannot, because when so many people are infected, the only thing you could do is what they did in the MS study, where they had EBV naive people, and they saw them become EBV infected over years because it was an Army study and they took blood every year from them, right? But they didn't do that here. Okay, so recent EBV reactivation is more frequent in women than men, and it's associated with a higher likelihood of severe COVID in men, but not women. So that's weird. Okay, in long COVID, being positive for EBV antibodies, reactivation was associated with a higher likelihood of post-inflammatory changes on chest X-ray. So this is, again, something that you can measure. And recent EBV reactivation was associated with a higher frequency of pathological values, okay? So you can measure some of these oxygen lung function tests that we talked about. And there's no signs of improvement over time in those patients. And finally, this is very interesting. Recent EBV reactivation was not associated with the presence of self-reporting symptoms. Okay? Which tells me that that's a mishmash of people just feeling bad and maybe some LC-specific stuff. IgA is the strongest serological marker of recent EBV reactivation and long-term pulmonary dysfunction. Antibodies, so these anti-EBV IgA were higher in individuals with long COVID than in the COVID-19 naive group. There was no association with pulmonary long COVID in people who had low levels of antibodies to EBV. They say this indicates EBV reactivation probably occurred months before SARS-CoV-2 infection. Okay, so it's not clear what's going on, but there must be something else in this equation, right? So they decide to look at specific genetic changes that could be associated with reactivation. And these are single nucleotide polymorphisms in the IL-1 gene cluster and the IL-10 gene. Right, so these are cytokines that are produced during infection. So they found an association between 4 SNPs, single nucleotide polymorphisms in the IL-1 gene cluster with recent EBV reactivation. Okay, so people who had recent EBV reactivation tended to have these single nucleotide changes. It's not 100% by any means, but it's statistically more frequent.
So it suggests that there are alleles, versions of these genes that may put you at higher risk for EBV reactivation.
Right. And there are a number of SNPs here. There are, in fact, 10 that they looked at. they have a higher representation in people with recent EBV than in those without recent EBV. So I just told you that, but that's another way to put it.
And these interleukins are associated with the innate immune response, right? So this is all making sense from that point of view.
It's an early response. The interferons are produced and cytokines are produced, including IL-1 and IL-10, that regulate inflammation, that induce inflammation. So, and in addition to people who had these SNPs in the IL-1 gene, we're still talking about, they're more likely to experience EBV reactivation during SARS-CoV-2 infection than non-carriers of the risk genotype combinations. All right. So another idea that these matter. So that's IL-1. Then they looked at another one, IL-10. They found no association with EBV reactivation with any of these SNPs. Okay. So they did find that one of this SNP in the IL-10 is more frequent in individuals with long COVID who have changes in the X-ray compared to people without the change, but no association with reactivation. Okay, so maybe 10 has something to do with long COVID, but not EBV. All right, so those are genetic predispositions. So then they said, well, what about the actual proteins, right? Do the presence of these in the blood correlate with the genotype? There is in the serum, there's an antagonist of IL-1 called IL-1 receptor antagonist, and they want to know if levels of that and IL-10 correlate with the corresponding SNPs that we have just talked about, EBV and long COVID symptoms. So serum IL-1 receptor antagonist in people with long COVID, no difference between people with or without EBV reactivation or with or without the IL-1 genotype. However, higher levels of serum IL-1 receptor antagonists were found in individuals with pathological changes on X-ray. So again, not with EBV, but may have something to do with long COVID. With IL-10, no differences in IL-10 in serum in people with or without long COVID, and similar to IL-1 receptor antagonists. Higher IL-10 levels are found in people that have lung changes after SARS-CoV-2 infection. They also look at circulating immune cell populations, and they do find elevated percentages of CD8 T cells in people with recent EBV reactivation compared to without. That's not surprising because you make more virus, you're going to have more of a cellular response as well. And they also find that a higher percentage of CD8 T cells in the subgroup of people carrying the high-risk genotype of IL-1 compared with those with the low risk. So, if that makes sense, if that is controlling EBV levels, right? And we'll talk about how that works in a minute. All right, what about the virus itself? They look at EBV in the blood using PCR and try and associate that with long COVID and antibodies, risk genetics, and EBV reactivation, okay? So people with serological evidence of recent EBV reactivation, which again, remember, you do by looking at antibodies, they have a higher level of virus in the blood. Makes sense, right? And in particular, the EBV DNA load in the blood correlates with serum IgA levels against the virus, right? A high DNA load of EBV in the blood correlated with pathological changes on chest X-ray or these reduced pulmonary functions that we talk about. And so this is an indicator of more severe pathology, having more EBV in the blood.
Right.
And those are the results, basically. That's it. So let's summarize them. So we see an association of frequent EBV reactivation with IL-1 genetics and higher EBV load, right? And so somehow, so just EBV alone, because they want to know what's causing reactivation, somehow IL-1 changes are affecting that, and you also get a higher EBV load in the blood as well. Serological reactivation was twice as frequent in long COVID, with objective signs of long-term pulmonary dysfunction. So again, the idea being there's a connection between EBV and long COVID, which is emphasized.
But that was a link only with pulmonary function in long COVID, not with the other long COVID symptoms.
Yeah.
Right. Not with the self-reported one.
Not with the self-reported one.
Exactly. Exactly right. Yes. And they say other studies have found in smaller cohorts, correlations, but ours doesn't show that because the self-reporting doesn't seem to be accurate.
Well, or there's something else going on with the self-reported symptoms that's not related to these cytokines.
So they say, there's something critical about EBV reactivation, either while you're having COVID, so the COVID could reactivate it, or shortly before or after infection. Okay. So these risk alleles, we know that this IL-1, for example, is a pro-inflammatory cytokine that recruits immune cells to the infection. And they say, okay, if these patients have dysregulated IL-1, then maybe they can't regulate EBV infection as well. That's why there's this correlation between those SNPs and EBV infection. Now, one thing that I didn't know is that there's some respiratory component to EBV infection on its own. And so they said we really don't understand how this works, but this could play a role in some of these pulmonary functions, right? So EBV could be a risk factor for long COVID because it's doing something in the lung. We don't know exactly what. And they say this needs to be studied, right? They say the strongest association of recent EBV reactivation was observed with what's called KCO, a parameter specific to the health of the alveolo-capillary compartment. And it turns out that the respiratory tract is a major reservoir for EBV, and so how it does pulmonary impairment is not known. That's an important thing to study. So people with risk genotypes leading to dysregulation of IL-1 have a higher likelihood of EBV reactivation during COVID-19, which leads to long COVID. And now this may not be the only risk factor.
Which leads to the pulmonary features of long COVID.
Pulmonary features of long COVID, right. But not to the self-reporting.
Right. And we're not certain that it leads to it. We just know it correlates.
Yes. We have a relationship between the two. Yeah. That's right. Because this is basically an observational study.
Yeah.
Right?
So EBV can replicate in both B cells and epithelial cells. Mm-hmm. I'm thinking about the lungs.
Yeah.
Okay. But, we've discussed enough about the immunology, the specialized immunology of the lungs. So it could be that even just infecting immune cells in the lungs could give you a lung pathology.
Yeah.
But it affects epithelial cells as well. So that's a possibility, it seems to me, as well. So we've all got EBV. I get COVID. That might trigger EBV reactivation.
Right.
That might be more likely to do that if I've got one of these mutations in IL-1 or IL-10.
IL-1.
And if my EBV gets reactivated, there's some association that might elevate my risk to long COVID. None of this is 100%, right?
Right.
But specifically lung pulmonary dysfunction.
Pulmonary,
right.
Yes. Right.
Lung COVID.
And it's just IL-1. IL-10 didn't really correlate.
Okay, fine.
Right.
Yes, lung COVID, which they can measure by X-ray and all these, tests where you measure pulmonary function.
They were very conservative in their conclusions, and I think rightfully so.
Yes.
I mean, they talked mostly about how this allows them to look at EBV reactivation and the role of IL-1 in that reactivation. And they didn't, they were pretty soft on their conclusions about the long COVID and everything else, rightfully so. But it's still very interesting. Very cool.
Yeah. I mean, their limitations paragraph is very good. They say all the things that, and they say, for example, we looked at IL-10 and IL-1, but there could be other genes involved as well. We didn't look at everything. We didn't look at saliva, which is typically where EBV is, because the samples weren't good enough.
By the way, as we've already discussed, all the authors here are from the Czech Republic. One of them from an institution in Brno, which will come up later in our discussion. That's where Mendel did his.
Yes, right. That'll come up during Rich's picks.
Oh, that's right. That's right. So the conclusion is the present examination of a large cohort of individuals exposed to the same trigger over a short period provides new insight into the biology underlying LC, but may be relevant for EBV-related diseases, such as autoimmune and inflammatory diseases and cancer, where sample size and unknown triggers make the analysis of large cohorts difficult. So that's what's unique about this. Okay, so I like it because it's a very data-driven study, where you can do x-rays and pulmonary function tests and say, this person has had long COVID in these tests for a long period of time. Okay. So our paper is from Nature Communications, bat-specific adaptations in interferon signaling and GBP1 contribute to enhanced antiviral capacity. Want to do the authors, Alan?
Sure. First author is Victoria Gonzalez. Senior author is RNJ Banerjee. And the team is from, of course, it's Nature, so you have to go to the end to find this out. The team is from University of Saskatchewan in Saskatoon, University of Waterloo in Waterloo, Ontario, Université Laval, Quebec, the Pirbright Institute in Surrey, UK, University of Cambridge, UK, Duke NUS Medical School in Singapore, University of Toronto, and University of British Columbia in Vancouver. So mostly Canadian team.
Nevertheless, with a serious international component.
Yes.
Mm-hmm. One of the authors is Lin-Fo Wang from Duke NUS, who has been on TWiV before.
Yep.
As you know, there are a lot of different species of bats all over the world, and they host a lot of viruses, particularly a lot of zoonotic viruses that have spilled over into humans, like SARS-CoV, SARS-CoV-2, MERS-CoV, and others. But they don't seem to get sick. At least we can't tell, right? They can harbor these viruses. For example, Nipah virus, which is lethal in people. The bats seem to be okay.
Rabies is another one that we talked about last week. They don't talk about rabies in this, probably among other things, because they can't do experiments on it very easily.
Right.
Mm-hmm. So why is this? If you accept that bats don't get very sick from all these viruses,
how do they do that?
How? Maybe that would be useful to know. Maybe it could help us, right? So in this paper, they're looking at the innate immune system, right? The first defense against infection, where sensors in the organism can sense things like nucleic acids, viral nucleic acids, and proteins, and these lead to the production of interferons, which in turn turn on antiviral genes. And there's a signaling pathway that's involved. The interferon binds to receptors on the cell surface, activates protein kinases, which phosphorylate a series of proteins, including two called STAT1 and 2, which are going to be the topic of this paper. And then eventually in the nucleus, you get transcription of interferon-stimulated genes. So they want to know if aspects of this pathway works in bats, because it hasn't really been studied all that much, right? So what they did here is to study two bats, two very different bat species, which is a good idea because you can get an idea of the divergence in whatever you're looking at. the insectivorous big brown bat, Eptesicus fuscus, and the frugivorous black flying fox, Pteropus electo.
And I'm wondering what Pteropus electo did to get named after one of the Greek Furies. I don't know how that happened, because this is a totally innocuous fruit-eating bat that hangs out in the trees and then goes and sucks on fruit. But
well, they're scary looking.
They're bats, they're big.
They actually look like foxes. The face is very fox-like.
They're flying foxes. One of the flying foxes. They are not the biggest of the genus.
I mean, if you have an issue with bats,
there's some big bats.
If you have, they're big. Okay, so if you got a problem with bats to start with, these are scarier than your average bat.
Yes, and Eptisicus it's called the big brown bat. It's not all that big because brown bats are small, but it's bigger than the little brown bat. So
I remember Jonathan Epstein, who's at the EcoHealth Alliance. They go do surveys of bats in various countries. They're interested in Nipah. And he has sampled them. You catch them, and they have a 3 or 4 or 5 foot wingspan. And he says, as you're holding them and you're trying to sample, they have these long wings with claws.
Oh.
And he said, you really have to watch out because they try and wrap that claw around and take your eye out.
Ooh.
Wow.
Wow.
Okay,
So
maybe the Latin name
careful.
does make some sense. All right.
Okay, so what they do here, they're focusing on interferon beta. Okay. We have a bunch of different interferons, alpha, beta, gamma, and lambda, but we're going to look at beta. And they make recombinant interferon beta protein. They make human, and they make it from these 2 bat species. So they clone the genes, and they produce protein and make antibodies to it, right? And then they say, does it work? So how do they tell if it works? Well, you use a virus, and you see if you add the interferon to cells, if it inhibits virus reproduction. And the virus that they use is vesicular stomatitis virus, which is a rhabdovirus related to rabies virus, because it's very sensitive to interferon. And so you can add interferon to cells, and then you infect, and then you measure the effect on yields. And they use cells derived from the bats, and they also use some human cells as well. And it works. It inhibits VSV reproduction in these cell lines. Okay, so that suggests that the interferon is inducing interferon-stimulated genes, because those are what actually inhibit virus reproduction. The interferon itself has no inhibitory capacity on its own, but it induces genes that are inhibitory.
So in each case here, we're talking about using the appropriate specific bat interferon on the cognate bat cell.
Yes.
And the human interferon on a human cell. We aren't messing around with mixing them up. That's too complicated. Fine.
But I think we will in a bit. Okay.
But initially, we want to know what they do in their natural context.
Right. So they have P. electo cells and E.
fuscus
cells, right? Yeah.
Yep.
Yeah.
Yes.
And they got, for human cells, they got A549s, which are a wonky cell that's been around for a long time. And
it's some kind of lung cell, right?
Yeah.
Lung cancer cell.
Riptic, and I'm not
as
familiar with that.
Seen that one.
That's a new one to me.
Or PTEC.
Yeah.
Yeah. What are these? Primary renal proximal tubule epithelial cells.
They're
Okay.
human?
Yeah, they're human.
Okay.
So both kidney and primary is significant. That means that if they're real primary, then, which I doubt.
No, I think this is a
then
line.
It's the first time they've been cultured. Okay. But it does mean that there are not many passages removed from their original isolation from the individual, as opposed to A549s, which God knows what's going on with them.
Have been kicking around in labs for a long time. Yeah.
All right. So next they look at antiviral efficacy with some different viruses. So they use MERS coronavirus, which is thought to have emerged from an insectivorous bat, not this one in particular, but an insectivorous bat. And so they treat the cells with their interferon beta, and then they infect with MERS coronavirus. And in the P. electo, the E. fuscus, and the RPTEC cells, they reduce viral load upon interferon beta treatment. And I presume, again, it's Rich is the homologous interferon in each case, right? Is that important? So they try it either pre-treating the cells or adding it a little bit after infection. And it works in most cases after infection, except in a certain human liver cell line. It only works prophylactically. So basically, they conclude that interferon beta can induce an antiviral state that is not antagonized by MERS coronavirus in kidney cells from all three species, humans and the two bats. So the reason they say that is because MERS coronavirus is known to antagonize interferon responses and blunt them so that it can replicate better.
In all these cases, correct me if I'm wrong here, but in all these cases that we've talked about, the bats do a better job of squashing the virus than the humans do.
That's right.
Right?
That's right.
It's statistically significantly different.
Yeah.
Okay, either in the amount of time it takes, or the effect on the titer of the virus, et cetera. So that's significant. The bats are stepping on it harder than the humans.
So these interferons work to suppress virus reproduction. Now they want to know if the signaling pathway from the cell surface to the nucleus is the same. And so this is called the JAK-STAT pathway. JAK is one of the kinases, and STAT is one of the proteins that's phosphorylated that then goes into the nucleus and turns on transcription. And the phosphorylation is done by tyrosine kinases. These are enzymes that put a phosphate on a tyrosine amino acid, right, at specific positions, very specific positions. So what they do is they pre-treat their cells, the bat cells and the human cells, with staurosporine. It's a broad-spectrum kinase inhibitor, so it's going to hit the JAK kinases that are involved in this pathway. And then they add interferon beta, and then they infect with VSV. So staurosporine is going to reduce the phosphorylation of STAT1/2 by JAK. And they show that by looking at phosphorylation status, that it does reduce it. It reduces nuclear localization because stats have to be phosphorylated in order to get into the nucleus. STAT1 and 2 dimerize after they're phosphorylated and they get in the nucleus. Remember all this, Alan?
Oh, yeah. Yeah, this takes me back.
It's old stuff. And then they also see that some of the interferon-stimulated genes, their production is reduced in the presence of staurosporine. So they're looking at the whole pathway. So basically, it looks like this phosphorylation cascade is intact in bat cells, and JAKs and STAT1 and 2 are involved. Then they look at the rate of phosphorylation and nuclear translocation of STAT1 and 2 after interferon beta. And in all the cell lines, the STAT1/2 goes in the nucleus within 20 minutes of interferon treatment. But the P electro cells, they get in there 5 minutes. They're really, as Rich said, stepping on it. Stepping on the accelerator pedal, right? 5 minutes, the P electro STAT1/2 gets into the nucleus. So it's kind of tempting to say that's part of why they do well with a lot of viruses, right? Because they respond quickly. So then they say, what if we take out STAT1 and 2 from these cells? They're not doing it in bats, they're doing it in cells. So they knock down the proteins with siRNA, small interfering RNA. So they knock down STAT1 individually and STAT2 and then in combination. The combination is lethal. The cells die. So apparently you need both.
You need one, yeah.
For life. But if you knock down STAT1, you reduce interferon-mediated protection. But in one of the cells, PACI-TO3 cells, but EFK3B and A549s were still fully protected from infection, even if you knock down STAT1. But if you knock down STAT2, everybody loses protection against infection, all the cell lines. So it's interesting. There's a differential requirement of STAT1 and 2. And then STAT1, 2 both together are lethal. So basically, that's what you would expect from human cells as well. Now, remember, MERS coronavirus can infect humans as zoonotic spillover from camels, but the virus is thought to have originated in bats. And it's been found by others that MERS coronavirus can antagonize interferon signaling in human cells. So they asked whether MERS coronavirus could do that in the bat cells that they have, whether MERS coronavirus could antagonize interferon signaling initiated by this interferon beta. So MERS infection alone suppressed the phosphorylation of STAT in the 2 human cell lines that they use. That's just confirming the observation. And interferon treatment alone induces phosphorylation of STAT against all cell lines treated with interferon beta, followed by MERS coronavirus infection. But if you treat the MERS coronavirus infected cells post-infection with interferon beta, you get reduced levels of phosphorylation compared to interferon beta treated cells. So the virus is antagonizing phosphorylation. And that's more of a natural world thing, right? Because the virus comes first and then your interferon goes up. You don't get pre-treated with interferon before you're infected.
The bats don't see it coming.
Well, yes and no. Because one of the deals, interferon has both autocrine and paracrine effects. Right? So you make interferon in the cell that has been infected.
Right.
And that interferon can stimulate the interferon-stimulated genes in that cell, but it also is disseminated to surrounding cells that are uninfected, sending out this danger signal and primes them by inducing their interferon-stimulated genes. So it's an early warning system, among other things.
Also, they do see it coming.
The basal levels
see it coming.
The basal levels of interferon can differ.
Yeah,
right.
You can have a higher basal level in bats, and I think at some point in one of these bat cell lines, they actually show that. So you treat bat cells with interferon beta post-treatment in bat cells. So in human cells, if you do post-treatment, it doesn't work. But in bat cells, it does. In bat cells, treating MERS coronavirus infected cells with interferon beta did not lead to a loss in STAT1 levels. So this suggests that the bat cells can resist the MERS coronavirus-mediated inhibition of STAT phosphorylation. So, and that is slightly different.
Which is consistent with the bats having lived with that virus for a while.
Yes.
But interesting that 2 different bat species from 2 completely different parts of the world. The big brown bat is a North American bat. The black flying fox is an Australian bat. Both are able to resist the MERS coronavirus shutdown of their interferon system.
So this is an ancient-ish bat thing.
It is. It is. All right, so then they look at the genes that are induced by interferon beta, the so-called interferon-stimulated genes. And they do that by transcriptome analysis of cells before and after interferon beta treatment. And they identify a lot of interferon beta upregulated genes in the bat cells that are similar to the ones that are induced in human cells, like ISG15, STAT1, IRF7, IRF9, and a bunch of others, TRIM5, all right? But they also find that some of the human interferon-stimulated genes are gone. For example, P. electo has lost a couple of these interferon-stimulated genes. There are also some induced genes that we've never seen before in humans. So there may be some novel ones in bats as well. And then there's some of these interferon-stimulated genes that are duplicated. Wow, maybe you do that to make more of it, right?
Yes.
And we don't know, but they said we could do experiments to figure that out.
That's important. Make copies of it. Yep.
In fact, I remember the first genome sequence of bats done years ago by Linfa and others showed that a lot of immune genes were duplicated, which they suggested could be part of their being resistant to infection. So they picked two of the interferon-stimulated genes to study a little more, IFIT1 and GBP1, IFIT1 and GBP1. And they take these proteins and they do some experiments that show that IFIT1 works similar to it works in humans. IFIT1 actually binds to the cap on messenger RNA, and it inhibits translation. And so they show that it works equally well as the human. But what they find different is these GBPs. These are guanine binding proteins that are actually conferring protection against a wide range of viruses and bacteria and protozoa. So interestingly, now in mice, there's a GBP cluster, 1, 2, 3, 4, 5. You can delete all of them, and it has no effect on virus infection. So in mice, they're not antiviral. In humans, they are. In mice, they're not. So they ask, well, what about bats? If they treat P. electo with interferon, it does not induce GPP1, but it does so in the other bat cells. They find that there's a motif in this protein that is important for the activity of the protein that seems to be conserved across the bat species. So they looked at the antiviral activity of this GPP1, humans, P. electo, and E. fusus. They produced the protein in human cells. They see that where it localizes in the cytoplasm, they see that it's active against a range of RNA and DNA viruses, but it differs among the different bat proteins. They look at herpes simplex, they look at influenza virus SARS-CoV-2 and MERS-CoV-2, and they see differential effects of the GBP from different bat species. And in fact, they look at a bat-derived pox virus, Epstein-Barr pox virus. This is for you, Rich.
Yeah, I got all excited about this.
GBP1 completely abolished virus reproduction at the lowest concentration, while the GBPs from human and the PA, the flying fox, were effective at higher concentrations compared to that one. So the GBP varies in its effectiveness against this bat-derived virus, depending on the species of bat cell.
And notice that Vaccinia just shrugged the whole thing off. Vaccinia is a pro at suppressing the interferon response.
Yes.
Matter of fact, there's some old experiments that address how Vaccinia does this that are based on the phenomenon that VSV, as you already said, is very sensitive to interferon. If you add Vaccinia to the mix, VSV grows because Vaccinia suppresses the interferon. It's really good at it.
Of course, Vaccinia didn't evolve in bats, so probably
no.
Didn't give the bats a chance to counter, right?
Yeah.
Then they also find a motif in GBP1 of E. fuscus, which is different and is essential for its antiviral activity. If you take it out, there's no antiviral activity. And so this is a different sequence, a different amino acid sequence that isn't present in human, which they're excited about, and they want to know what it does. So essentially, what they have shown here is that interferon beta induces this phosphorylation pathway involving JAK and STAT proteins. It induces interferon-stimulated genes that can inhibit viral reproduction. You have differences among the two bat cells in the levels and the speed. They say the E. fuscus-derived kidney cells express basal levels of nuclear STAT1/2, which suggests that they may be primed to respond faster to interferon. So there are these little nuances that may be consistent with why bats are more resistant to pathology, right? And the JAK-STAT pathway is not antagonized by MERS coronavirus during infection in these two bat cells. So that's good for the bat because that means they can reduce MERS coronavirus infection through this pathway. Yeah, so that's it.
Now we need humans to be able to pull off this trick.
So, I have a couple of comments here. First of all, I have to say that when I was reading this, and I just said that I got all excited when I saw pox viruses. And I'm amused at the nerdiness of that, that you actually get really emotionally wrapped up because your virus is on stage here. And ooh, what's this about? God.
It's like you're there with one of those foam #1 figures. Yeah, right. Pox.
Yep.
So the other thing...
holding a lighter over your head. Yay.
That's it. The other thing that I'm having that I was in my head the whole time I was looking at this. So the fundamental problem here, or fundamental issue, is that what we say is that bats harbor these viruses and they spill over. So I'm keyed on, and so we have evidence here that the bat's interferon system is behaving in a fashion that would make them more resistant to these viruses. Mm-hmm. On the surface, that almost seems like a disconnect or a contradiction. Okay, that if the bats are more resistant to these viruses, they wouldn't harbor them. They wouldn't have them. But that's not what we're talking about. So it seems to me that the issue here is that the interferon response makes it so that the viruses don't necessarily kill them. Right? But does that mean that I don't think this necessarily means that the bats harbor like a persistent infection with these viruses.
Right.
I wonder about this. Is this, do viruses carry these around? I mean, do bats carry these around long term, or do colonies, as a population, harbor these viruses, where a few individuals are infected maybe transiently now and then? So a colony can support replication on a population basis of this virus for a period of time, because it's not going to kill them. All right.
My understanding is that that's what you just described is going on. Okay. That it is spreading in the bats with limited or no pathology, and it just circulates among them. Right. They're not persistently infected. I'm basing this on what I've seen. Usually a lot of studies have been done on rabies in bats, and different bat species have different rates of rabies infection, but a lot of bats are negative for the virus but positive for antibodies.
Yeah.
Okay, so they've seen
Which is not something you see in humans unless they've been vaccinated, because if you get rabies, you die. So bats are apparently getting rabies virus, surviving, building antibodies, but it's passing around, and different bat species vary in their frequency of infection with rabies virus. But a lot of them are flying around with it, and spreading it. And I believe it's the case with many of these other viruses, too, that they are spreading in the bat population and not killing the bats.
Okay.
But not persistently infecting.
And rabies is a great example because, as we discussed last week, in all other creatures that I can think of, it's a lethal infection.
Yes.
It kills you. Okay, so you're not gonna support that over a long period of time in a bat colony. It's gonna wipe you out. So that's a great example. The colony will support this infection over time, and not every bat's going to have it. You just if you get infected as a human from a bat, you were just unlucky enough to catch the dude, to interact with the dude who, at that period of time in the colony, was the one having an active infection.
Yeah.
Who would probably clear it ultimately.
Yeah, it's like rhinoviruses or flu with us. It circulates, but we're mostly surviving it.
So, for example, rabies-related viruses, only 1% of colonies carry the virus at any given time.
Okay.
All right. So I remember Jonathan Epstein's study of Nipah virus in flying foxes. He found that there are pulses of virus production that correlate with stress, right, or pregnancy. So maybe you have a low level or none, and then you get virus in your colony, and then you have a stressor, and it goes up, and that's where it's transmitted from those stressors who are making a lot of viruses, so the stressed bats. But yeah, in any case, they don't get sick, probably because the interferon system is keeping the levels down.
Yeah,
right?
So I'd like to see some sort of statistical or epidemiological study that correlates the resistance to the virus to the persistence in the population. Does that actually make sense? Okay, intuitively it kind of makes sense. I'd like to see the analysis.
Yeah,
it's probably out there.
Mm-hmm. No, I think Epstein at EcoHealth has done a great deal. We should get him on and talk about it.
Yeah.
The one thing I like about this is that it shows there are differences between humans and two different kinds of bats in these interferon responses. So you can't, do some experiments in cell culture and say this is how it works everywhere.
Right.
And most people use human cells anyway, but it's different.
And you can't treat an order as if it was a species.
That's right.
Which is, it's kind of funny that that's one of the main points of this paper when their first sentence is, bats are one of the most abundant and geographically diverse mammalian species in the world. And I highlighted that because I was like, no, bats are not a species. Bats are a whole bunch of species.
Let's do a couple of emails. Let me take this first one. Mary writes, here's a chuckle to go with your morning. Seattle Times editorial cartoonist David Horsey hit the nail on the head when he notes it is undetermined whether any of the new committee members are witches, warlocks, fortune tellers, or snake oil salesmen. This is the ASIP panel put together by RFK Jr. It's a cartoon.
Yes. Showing a bunch of witches around a cauldron.
Alan, can you take the next one?
Yes. Alex writes, Dear TWiV team, being a huge fan of the podcast and a new dad to a little girl, I've been unsuccessfully trying to find the somewhat recent clinical update episode that featured a study on the immunologically optimal timing of the first measles shot. If I remember correctly, it was something like 14 months, but I'd like to read up on it again before talking to our pediatrician. I would be super grateful if you could point me to the right episode. Thank you so much for everything you do, and sending our regards across the pond all the way from Germany. And Vincent, I guess, has put in the TWiV 1117 is the episode you want there.
So we covered it twice, on TWiV and on Clinical Update. So I put the TWiV, because we did a deeper dive.
Yes.
Yeah, I actually went back and looked at this a little bit, at least the first part of the episode. It actually discusses a paper that addresses this very issue, and it does it very thoroughly, and it's really good.
Yeah, I think they concluded around 12 months was optimal, not sooner.
That's what's used, and that's what's used in the U.S. for undoubtedly these reasons. One of the things that was emphasized, however, in the paper and in our discussion is that there's a lot of individual variation.
Yeah.
Yep. It's always a problem, right? You have to make a decision for the population.
Yep.
Rich, can you take the next one?
Anthony writes, insurance companies, HMOs, and even government entities must be distressed that what for a century has been a predictable/negligible and so trivially manageable cost will be lost. The replacement will be much difficult to forecast and highly expensive illness. What are we talking about?
I think vaccine preventable diseases.
Vaccine preventable diseases. There's also soon to be proposed a prospect of outbreaks on a scale of natural disasters. Before 2020, healthcare payers never had a plan for claims on the scale of a hurricane or a tornado. Are healthcare companies doing anything to counter anti-vax? I don't know about it. Thank you, Anthony.
Good question.
So I don't know, actually. There's a lot of certainly scientific establishments and scientists and people concerned about healthcare, not necessarily in the business, who are raising alarms.
I think it's problematic for the companies. Well, I guess the healthcare companies, it's not. The manufacturers, it would be, because they would just say you're conflicted.
Yeah.
I don't know. Good
but
question.
it's a highly centralized industry where a lot of the healthcare companies are also involved in vaccine manufacturing.
Yeah.
We're doing a lot of controls here, gang.
Yes.
Peg writes, hi, TWiV, great podcast. Some news about the lack of vaccine uptake is always important. However, as a fully vaccinated senior, I'd like to hear more about how I can protect myself from the unvaccinated, given that they may be spreading a dangerous virus even before they know they are ill. I live in New York, but I visit my adult children in Pennsylvania. Well, vaccination is a great first step.
Yeah.
But if you are worried, you could always wear masks when you're in public among a lot of people. And that helps as well. Or you could just stay home. But that's not really
that's not much fun.
So vaccinating and masks.
You know, it's interesting because in a way what she's talking about here indirectly is herd immunity.
Yeah.
Okay, because ideally everybody ought to be vaccinated against the diseases that are going to cause a problem.
Alan, can you take the next one?
So,
Mary.
One of the things she ought to do is vote.
Vote. Yes. Although from she says New York State, I'm not sure where about, but to adult children in Pennsylvania, that could be a long boat trip. Which is not necessarily a bad thing. Just all right, Mary writes, Dear TWiV team, I am a regular listener and understand you are very concerned about public health as it is impacted by loss of trust in science, vaccines, and the healthcare profession. Recently, I met the mother of the lead author of a paper on this subject. The paper identifies a need for data on individuals' levels of trust and their subsequent healthcare access, using data from the Pandemic Recovery Service conducted across 21 countries with over 621,000 respondents. The authors examined the relationship between trust in healthcare professionals and healthcare access delays. By understanding how trust influences healthcare access, the study seeks to inform policies that improve health outcomes and address the complex interplay between trust, access, and quality in healthcare systems. Provides a link. Also, another awful tale of the current administration's efforts to destroy trust in the system appeared recently in the Seattle Times. Washington immigrants' protected health data was shared with immigration officials. In this case, it was through the state's expansion of Obamacare, a favorite target of the current administration. And provides the article. Be interested in seeing somebody go after that as a HIPAA violation? Just putting that out there. I have no idea what the legal ramifications of it would be, but that just strikes me as probably something you could at least sue over. And Mary continues, of course, these assaults on the privacy of immigrants make me and everyone else less trustful of the healthcare system. Hopefully the paper demonstrating the relationship can be used to inform a counterattack. Anyway, as soon as my new friend told me about it, I knew you were the folks to share it with. Onward and upward.
Mm.
Thank you, Mary.
So this study that she cites to start with about people's trust in healthcare, these are some of the types of studies that the current administration has made efforts to defund.
Yes.
Because they don't want to hear about it.
Yep.
Rich, you're next.
Peggy writes, I saw this on BBC and thought you should see it regarding the discussion on rabies vaccination. And the title is Britain Dies from Rabies After Scratch from Stray Puppy in Morocco, BBC News, and gives a link to the news article. We mentioned this last week when we discussed rabies. Man, a scratch from a puppy, right? Who would have thought? I haven't actually looked at the article, but
yeah, that's basically it. So if you're scratched by a wild animal, get a rabies vaccine.
Yep.
Daniel writes.
I'm wondering how long, if it says.
And
I think she was dead in 2 weeks.
Quick.
And this was a puppy, so not a wild animal. But if you're in a place where vaccination of pets is not routine, count that as a wild animal.
Yep, absolutely.
It was a stray dog, you know.
Yeah, stray dog. They're the major suspects.
Yeah.
Okay.
So she was bitten 2 weeks ago. Was she? I think it was 2 weeks. Wow, that's fast. Okay, where are we? Okay, Daniel. TWiV 1227 had a listener pick on x.com that the hosts couldn't view due to not being logged in. You can fix that by replacing x.com with xcancel.com in the URL.
Uh-oh.
I didn't know that. You can read about their paper for more info and browser extensions to do that automatically. xcancel.com/about. Thank you, Daniel. That's useful. And then we got another one from Anne. I found this link posted by a thread reader app, which will allow non-X Twitter users to see the thread you'd mentioned at the end of 1227. To me, some of the videos seem to have been crafted for clicks. Also, in reference to Nobel Prize winners making claims outside of their fields, and hence they shouldn't be assumed to be experts outside of their fields. There are perhaps too many examples of that. Two that come to mind are Linus Pauling, a brilliant chemist, biochemist, and his vitamin C claims about cancer and the common cold, and William Shockley, the transistor, who, after he retired, lectured on racist and eugenicist topics. There's a section on his Wikipedia page about this. Wikipedia also has a page listing other Nobel Prize winners who have what has been defined as Nobelitis.
Nobel disease.
Oh, this is great. This. Very good. I like that. An
informal term for the embrace of strange or scientifically unsound ideas by some Nobel Prize winners, usually later in life. I'm glad this is a document. This is like a diagnosable phenomenon now.
Very good. Very good. So like nobody's immune, right?
Yes.
Feynman, right? The easiest person to fool is yourself.
Yep.
Alan, you want to take the next couple?
Sure. Bill writes, just sends a link here. It's a science alert. Here's why rational people skip vaccines even when they trust science. And I have not read this article.
This is an article which says it involves game theory. Game theory reveals that vaccine hesitancy is not moral failure, but simply the predictable outcome of a system in which individual and collective incentives aren't properly aligned.
Well, the problem with game theory, which is an interesting academic exercise, is that it tries to reduce, when you apply it to things like this, you're trying to reduce hugely complex human decisions to what game theory loves, which is two-player zero-sum games. Game theory is all over two-player zero-sum games, but life is neither two-player nor zero-sum, and so I'm hesitant to embrace the logic of an article that applies it to something like vaccination decisions.
Yeah, I understand. Anyway, this if you're interested, it's called Game Theory Meets Vaccines.
Okay.
And they have a polio poster in it, Alan, that is reminiscent of the one that you had.
Yes.
Remember?
Yeah. Yeah, if polio comes.
Do you want to take the next one too?
Yes, right. That was a short email. Jay writes, hope this brightens your day. Headquarters for vaccine. This is from the Herald, October 12th, 1897. A very small ad. Just says, headquarters for vaccine. We are the sole agents for the celebrated New England Vaccine Company's virus. We sold during the last epidemic 90,000 in this city. Fresh daily. W.H. Chapman, 2637 St. Catherine Street. They give their telephone number at 4067. And
Rich, do you think?
Guessing is this the New York Herald or St. Catherine Street? Where is that?
Could be. Rich, do you think this is like smallpox vaccine?
I was thinking the same thing. It's got to be smallpox.
I mean, 1897, what else is it?
Yeah.
Well, when was the rabies vaccine developed? But these guys wouldn't be distributing rabies.
No, they wouldn't be distributing 90,000 of them during an epidemic.
That's got to be smallpox vaccine. And this immediately makes me think of, who's the guy? Jose Esparza, who collects old vaccines from all over the place and compares them. Well, we really need to update this story, okay? Because I wonder if these guys, I wonder if there's any remnants of these guys around. They still have some vaccine.
Huh. It's funny. Fresh daily.
Fresh daily.
Just scraped it off the cow.
That's what we need. Actually, that's probably not irrelevant, right? Because that was before freeze drying.
Yep.
So fresh daily may have some significance.
Yeah.
Who knows?
All right, one more, Rich.
Chris writes, hello, Vincent et al. Just wanted to reach out and say thank you for all the great content over the years and helping me stay informed on the topic of virology. I'm emailing you today because I often use episode 496 as a resource to show people that are vaccine hesitant or when debating anti-vaxxers online. The episode is truly one of the best resources for combating vaccine misinfo. However, there's one problem with the episode. It is about 7 years old and is pre-COVID. I personally would love it if you could make another episode akin to 496 just to update some of the current talking points and add to info about COVID vaccines and the mRNA vaccine. Also on the show, it is often mentioned that the very best way to fight anti-vax is sometimes with anecdote. So here's a discussion line that I found very effective for combating anti-vax, vax hesitant. Me, do you know anyone who has died during COVID? Almost everyone knows at least one person who died during COVID, not even including high-profile deaths. Them. Yes. Me. Well, personally, I know of at least three people who died directly from the virus. One was a guy in his early 50s and was unvaccinated. However, I don't know a single person that even had a bad reaction to the vaccine. In fact, I've never even met a person who says they know someone who had a bad reaction to the mRNA vaccines beyond a sore arm and maybe grogginess the following day. Hell, even beyond death, I know a few people that had long COVID from vaccines, with one of my friends still saying his sense of taste is 90% of what it once was. But I don't know anyone who had that after the vaccine. He means long COVID from the virus.
From the virus.
The virus. I don't know anyone who had long any of this after the vaccine. Chris continues, I have found this to be extremely effective approach for discussing vaccines with people. Granted, I haven't used this against a hardcore anti-vaxxer. However, with people that have more grounded skepticism, it works well as it really puts into perspective how the fake alternative news doesn't align with their actual experience, but their personal experience does actually align with scientific consensus. Amen to that. Also, you gave me a shout out of TWiV the other day, so I just wanted to say thanks. Thanks for your time and all the great info, Chris.
I just want to. My only suggestion, Chris, is that you slightly modify. I think you've got a great pitch here, and that's very similar to what I say to people who are vaccine hesitant. If this comes up, who do you know who's gotten really sick or died of COVID, and it's very true that most people have some story of somebody, either primary or secondary acquaintance. However, I know of personally in my immediate family 3 people who had horrible reactions to the mRNA vaccine, including me. Very unpleasant experience, still way better than dying. So I wouldn't go too far out on the limb of nobody ever has a bad reaction to the vaccine. A lot of people do have worse reactions to the mRNA vaccines than they would to a flu shot. I mean, I get a flu shot, I barely feel it. I don't even process, any kind of, my shoulder might be slightly sore the next day. My COVID-19 shots have been rough. But that clears up within 24 hours.
Yeah.
I feel awful the next day, and then by the next night, I'm fine.
I know people with a similar experience that was fairly long-lasting, and I noticed recently that, I don't know where I dug this up, but that the most recent formulation of Moderna's vaccine is 1/5 its original dose.
Yeah.
Okay. Because I think that was a significant difference between the Moderna vaccine and the Pfizer vaccine. There's other differences as well, but the Moderna vaccine was, I think, more problematic than the Pfizer vaccine.
Okay.
With respect
the one I got initially was the Moderna. I did get my 4th, I think it was. So I got the 3-shot series of Moderna, and then a couple of years later, I went ahead and got a booster. And that was the Pfizer. And the Pfizer also was a little rough.
So this episode 496. Vincent,
but I still highly recommend getting the vaccine.
So, Rich, that's the one you and I did at Irvine.
Yeah, I remember that. So that was Bert Semler ran that. Did we get invited to do that? Was that a special deal? It was an Irvine sort of in-house convocation.
Vaccines in the 21st century was, I think, the...
Yeah, he had put together a couple of people who had vaccine experience. And also Stacy Schultz-Cherry was there.
Yeah.
Right.
And 2 guys. It was good.
Yeah. And I guess, this is an example where something that might have had limited exposure otherwise got TWiV'd.
Yeah.
And we can help broadcast that message. I'll listen to this again. That was pretty good.
Okay, time for some picks of the week. Rich, what do you have for us?
Okay, let me scroll down here, but I know what I got. Apparently, we were discussing in the pre-show, and from your notes, my pick has been picked before on TWiV and also discussed on TWiVo. But Alan told me we ought to go ahead and do it anyway. It's there's a summary article that, where was it? I chose the PubMed link so that you can at least read the abstract because it's behind a paywall. This is in Cell. It's a summary article entitled Unraveling Mendel's Final Mysteries, a Genomic Retelling of P Genetics. And I'm also linking here the original article that this summary article is based on, which is also is a Nature article by a big group in China that did genomic sequence on almost 700 pea plants of various varieties and nailed down at the molecular level the genotypes of the 7 characteristics that Mendel studied to do his P genetics. So this is, and the interesting thing is that I didn't go through all the genotypes in detail, but they're all different. There's translocations, there's transcription factor mutations, there's transposon insertions. I mean, there's an incredible variety of differences. Okay, so this is what 175 years later, about something like that. So I just think it's just marvelous to look at this whole thing come, I would say full circle, but I don't know what's going to happen next. Okay, it could be that there's more down the road. This guy, Kathy Spindler, showed me these articles. And I said, ooh, TWiV pick. She says, you go ahead and do it. Because she knows I like Mendel. I taught genetics for a while in my career, and even to first-year graduate students who'd all had it in high school and probably college. I'd go all the way back and do Mendel and do Punnett squares and everything else, because every time you think about it, you learn something new. And that got me into a deep dive in Mendel, which I will circle back to because I'm reading a biography, and I will doubtless pick that when we get to it.
So originally, Alan had picked this on TWiV.
Yes.
And I said, whoa, we got to do that on TWiVo. So Nels and I did it on TWiVo. And now you're bookending it with another pick.
I'm patient zero of this pick. Yeah. And the Nature paper, the primary source is open access.
Oh, it is? Good.
Yeah. So you can actually skip the summary that was in Cell and just go directly to the Nature article if you want to download that. But it's also been reported. I think I picked a Science News article that described the results as well. And then the Nature article.
You know, as I'm sitting here thinking about it, it took Mendel 7 years to do his studies. And the book that I'm reading describes in detail how you actually do the hybridization, technically, of the peas. And it's an incredibly tedious prospect. And he did tens of thousands of crosses. And these guys sequenced nearly 700 genomes. I wonder how long that took them. Probably not 7 years. Certainly not 7 years.
You got to divide by the number of authors and find the hours per person.
True. Mendel was a sole author publication.
It was one guy. Yeah.
Yeah. Okay.
It's a very cool paper. Yep. Alan, what do you have for us?
I have winners of a photo contest. Nature has their Scientist at Work photo competition. Field biology, of course, rules this competition, but that's fine. They do hard work, and they ought to get some credit. So these are the 2025 winners, and they are some awesome photos, and they give the story behind each one. And I recommend scrolling on through here.
Dixon would like this.
Yes, Dixon would love this.
I think my favorite is the two ice core researchers who are drilling a core out it's polar nighttime in Svalbard, and they look like they could be on Europa or something. They're completely, covered up in parkas, and they've got their headlamps on, and they're handling this sample into a bag. I'm like, wow, that's where the aliens are.
I like the woman with the frogs.
Yes, that's
she's got this incredible smile on her face.
Yes.
Like, what could possibly make me happier than a handful of frogs?
A double handful of frogs.
Cute.
Yes,
very cute. My pick is a Nature article by Smitri Malapati. Signs of AI-generated text found in 14% of biomedical abstracts last year. Oh my gosh, people are using AI to write their bloody papers?
Yep.
Come on, folks. I mean, writing is hard. I remember writing many things, and you just do it. And in the end, and you do it over and over, you rewrite and you rewrite, and in the end you have something that communicates. Why would you want to let a machine do it for you? It's actually fun, and you learn a lot doing it yourself.
You would let a machine do it for you if you are under pressure to earn tenure by getting a lot of publications, and so you're pumping them out through a paper mill process, and you simply can't do that physically with the amount of time available and generate, the vast quantities of chaff that's being blown into the scientific literature these days.
So I have a love-hate relationship with writing, okay? And I want to bounce this off you, Alan, because there's a phenotype that loves writing that I just don't understand, okay? So the activation energy to get me to write something was always absolutely enormous. But then there would be times when I was doing it, when I would get into the weeds and start messing around with sentences and paragraphs and saying, how do I want to say this? And then looking at what I'd done and thinking, oh, that's not half bad, okay? So I could sort of get just a little bit of a taste of how you might actually be motivated to do this, okay, on a regular basis. But as I understand it, there are people who, like so many other avocations, can't not write, okay? They just have to do it. So what's up with that?
And writers actually fall into both these camps too.
That's interesting.
And everything in between. Writers are not all people who just, constantly write and love to do it from the get-go and, can just slip right into it. I have some activation energy myself. There's a hump you got to get over. And then once I'm into it, and with me having done this for almost 30 years, as my main line of work, it's kind of like, okay, let's get going. Yeah, now we're going. It's more of a speed bump than a wall.
Okay.
Then there are people like, I presume Stephen King, who just like words, it's just, if you look at the guy's output, and there are others who have produced even larger quantities of saleable prose in their lifetimes. And it's like, my gosh, how do you do that? So those are the people who have little or no activation energy. They're writing. But if I go for a while without writing, I do feel like something's missing. So in that sense, I do have to, I feel like it's something that I need to do. And that certainly helps with the speed bump. But your experience of it, that it takes a lot to get going, is very, very common. And there are professional writers like that too, who tend to produce less for obvious reasons, because it's very hard for them to get going. But in some cases, they'll have like one or two great masterpieces they produce in their lifetime, and that's their entire output.
Joseph Heller.
Yes.
Catch-22.
Yes.
That was it. That's what he wrote.
He wrote a sequel to that. It wasn't a sequel. It was another
He wrote a
book.
few things, but
Catch-22 was brilliant.
Yeah, that was his big thing.
Well, this amazing 200,000 abstracts out of 1.5 million in PubMed contains words commonly suggested by large language models. So that's how they do it. They find words that are used in excess of what normal humans would do, like heighten, hinder, unparalleled, and invaluable. Man, they sound pretty innocuous to me.
But if you've got them all in one abstract, it's kind of, what are you doing here?
Yeah.
So there's an error bar on this. They didn't actually confirm that all these were produced by AI.
Of course.
But they're finding hallmarks that some non-trivial proportion of the scientific literature is probably being written by robots now.
Now, there are programs that look for that, right?
Yeah.
You can take an essay and put it in. They'll say,
teachers use this all the time. Yeah.
Yeah. Well, it's kind of interesting what we're going through here.
Yeah, here we go.
Mm-hmm.
Yep.
All right, that'll do it for TWiV 1233. You can find the show notes at microbe.tv/TWiV. If you want to send us questions, comments, or picks of the week, TWiV at microbe.tv. And if you enjoy these programs, we'd love your support. microbe.tv/contribute. Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
I'm Vincent Racaniello. You can find me at Microbe TV or on Blue Sky, microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.